In order to test the effects of antibiotic resistance, we will expose 1 tube of E. coli to ampicillin, which is an antibiotic. This should kill off a large number of the E. coli and leave only those who are somewhat resistant. In another control tube, we will have a population of E. coli that is not exposed to the antibiotics, so they're not expected to undergo any antibiotic resistance. The dependent variable for this lab is the number of antibiotic-resistant bacteria. The independent variable is whether the population is introduced to the antibiotic or not. At the end of the experiment, we will compare the end colonies to the beginning colonies, as well as the colonies that were treated with AMP to those that were allowed to grow without antibiotic resistance. Bacteria evolve resistance to antibiotics because it acts as a strong selective force. Antibiotics kill off a majority of the population, but the bacteria that have even the smallest amount of resistance or beneficial mutations will survive long enough to reproduce, and thus increase the frequency of resistance within the population.
E. coli is a type of bacteria commonly found in the human digestive system. It is used to aid digestion as well as protect against certain harmful microbes. E. coli is particularly important in scientific research and has been used as an important tool in the research of genes and gene regulation. Its genetic flexibility and fast reproductive time make it a great tool for scientists. Ampicillin is a medicine often used to stop the growth of bacteria. It is classified as a penicillin medication, and it can be used to treat several infections caused by bacteria, including GI infections caused by E. coli. E. coli's resistance to ampicillin is increasing. However, as ampicillin works by stopping the growth of bacterial cell wall, E. coli are becoming resistant to this as they reduce the permeability of their outer cell wall and increase their drug efflux pump that is used to remove the harmful ampicillin and therefore decreasing its effects on the bacteria.
At the end of the experiment, when the tubes without AMP are put onto plates, one containing AMP and one containing just LB, we expect that there will be no colonies or very few on the AMP plate because they are not resistant to the antibiotic. On the LB plate, we can expect that lots of colonies will grow because they can reproduce and grow without interference. When the bacteria in the tubes containing AMP are put onto the same plates, we can expect to see the plate with AMP to have lots of colonies because the bacteria have become resistant and can still grow even when they are exposed to the antibiotic. On the plate with just LB, we can also expect to see lots of bacterial growth because they're still capable of growing in their normal environment. When we compare the two LB plates, we can expect to see more colonies on the plate that has bacteria that were not exposed to AMP. This is because the bacteria are not resistant to the antibiotic, and those that were resistant to the antibiotic have to use lots of energy to maintain their new adaptations, likely decreasing their ability to reproduce and grow as the same bacteria that never evolved antibiotic resistance.